welcome back for our next to last lecture. So yeah, chapter 4 is our last set of lectures. Yep, I said chapter 24. I didn't say chapter 4, right? Okay. Anyway, oof, slip of the tongue. Anyway, but yeah, oh my goodness, you've made it. You've done it. You've survived. You've done it, guys. Awesome work. So me and Monty are so proud of you. Except I don't know where Monty is. Oh, here he is. Hold on. Come here.
Huh? What?
Come here. Tell everybody how proud you are of them.
I am very proud.
So, yeah. Well, let me go ahead and hit this. There we go. No more bunky noises. Now, you may hear my son in the back a little bit, but other than that. So, yeah, we've done it. We've reached the last chapter, which means the week after next we're going to review a little bit. I'm going to be putting up some more information about how that works. Sorry the lectures were a little bit late. I was kind of going through some things, trying to get some things straightened out on my end, so I do apologize for that. Your final exam, so let's do some cleanup stuff. Your final exam will be 75 questions. Those questions will be 25 review questions from all the quizzes that you've been taking out throughout the semester, and then you will be, hold on, using all 3 of my brain cells. And then the 50 questions will be on chapters 23 and 24, if that makes sense. So you will have a review like we have been doing, where I just give you the words and you just study up on those and go ahead and take the exam. And number twosies, the 25 questions, I will have a quizzes link up, and I'll show you how to do that. I'll run through it for you with a little video for next week, showing you what to do and how to study for the final exam. The final exam will open on July 7th and then shuts on July 12th. So you got to get in there and get her done. Hi, yes, hello, how are you doing? Okay, sorry, Monty wanted a kiss. And I'll talk more about that next week, so that way I'll explain how everything's going to go down, so that way hopefully you don't have too many questions. Sorry about not putting the forums up from last week. I don't know where my brain went. Anyway, with that said, let's go ahead and get into chapter 24, shall we? Our last chapter. Oh. Alright, so last chapter, honestly, is pretty cool. I enjoy talking about this chapter. Of course, I enjoy talking about the majority of the chapters in anatomy and physiology. I enjoy them muchly. So, that is genetics and genomics. Now, this field is constantly changing, which is probably why they have to update the anatomy and physiology book so much. Because honestly, other than everything else, with the exception of a few rare things, we've mostly got everything down pat, sort of, kind of. So, but the genetics, oh god, this is constantly moving. This is one of the big movers in the biology world, is definitely genetics and genomics. Because it's changed so much, even from when I was taught it, to the fact that I actually probably should go back and get myself a refresher on some higher end things, because some things have changed. Actually, a lot of things have changed. Anyway, so genetics is the study of inheritance and characteristics in human variation, and genes are DNA sequences that encode for proteins that make us up. Now, I edited this and I wanted to kind of show it to you just how much it changes just within the span of 10 years. About 1% of our DNA codes for the proteins that make us. The rest of it is called the exome. Now, a long time ago, we used to think that was junk DNA. It is not junk DNA. It is not. That's actually something I had to update myself on. This is just how quick genetics changes from year to year to year to year. We're always finding so much more new stuff. The exome, just because we don't use it, doesn't mean it's just junk DNA. Actually, a lot of it carries over to our kids. A lot of it is used down the road, genetically speaking, and a lot of it is actually turned on and off, which brings us to the study of epigenetics, which is how our environment actually turns our DNA on and off depending on what's going on. And we've been finding this definitely going into space. I've been mentioning periodically throughout this series, how our body responds to being in microgravity because we're, evolved in a planet with gravity, and then coming out of gravity kind of mucks us up a bit, including our bones, our circulatory system, things like that. It's actually kind of weird. And if you haven't taken, 168 with me, and I believe most of you have, and have not, but and when I talk about the bones in 168, one of the crazy things is the osteoclasts and the osteoblasts. The osteoblasts make new bone, the osteoclasts break down bone. For some reason in microgravity, the osteoclasts go crazy and just break down your bone. But and your osteoblasts, which should be triggered and turned on to come in and form new bone matrix, for whatever reason, when it's in microgravity, goes, I don't want to. And doesn't. And that's actually why astronauts lose a ton of bone mass while they're up in space. That's why they're always exercising. And that's also why they actually get pumped full of osteoporosis medications. A lot of osteoporosis medications get pumped into that before they go up to try to mitigate a lot of what they lose in space. So even with constant exercising, which is our only other way to try and fight off bone loss. So it's weird. And so NASA did twin study, a very famous twin study, where they sent two identical twins, one, brothers that were in the Air Force, and they sent one up for almost a year, hair under a year, with a Russian colleague. This was before all that insanity that's going on in the world today. But, when NASA and Russia were actually working together, which was nice at the time, but now it's like, anyway. So cooperation, guys, honestly. So going up into space, we shot him up, and we kept his twin, and we took both their DNA samples before we shot the one up into space on the International Space Station. And then when he came back down, we instantly took their blood again, because we used his other twin as a control group, and him, just to see the differences of what in his genes got turned on and off by being in a microgravity area. And the studies are coming out, and it's absolutely fantastic. So we are trying to figure out more of what happens when we try to go into space or try to go into environments that are not the norm for us, like underwater and whatnot, because we know less about underwater than we do about space. Space is easy. Underwater is hard, as recent events have shown us. So we're still learning so much about DNA, and it's changing a lot, including the story I'm going to tell you next, which is just a quickie story. So genetic info is passed from generation to generation via meiosis and fertilization. Genetic functions at all levels of biology, which is biochemical, cell, tissue, individual, family, population. Genetics has its fingers in everything, because genetics is pretty much the basis for everything. And the study of it is intense. Like I said, I had my genetics courses 20 years ago, and the things that have changed since then, since I was originally taught back in college and grad school, oh boy, they've changed. For instance, our guy here. So this is Gregor Mendel, and for those of you that have taken or even, smidgenly look into biology before, Gregor Mendel should be kind of a familiar name. He was actually born when people throw out years, and I've always been one that I've never understood when you throw out a year and you're like, because that year doesn't mean anything to you unless you've been living in that said year, right? And even then you kind of lose things looking backwards, which is probably why people get rose-colored glasses about the past. Anyway, but he was born Johann Mendel in 1822. Now in 1822, we were going through our Civil War. So if that gives you kind of an indication of what time period we're looking at Civil War era. But this was over in Europe. So he was in what's now the Czech Republic, but back then it was not. So, and I'm not that good on European history studies, especially the middle of Europe, where there's a whole bunch of weirdness going on from, decade to decade. So, anyway, but he basically joined the church because he was really good at school, but he was the second son of a poor farmer. And the only way he can continue his education was to become a member of the church, because they sponsored a lot of their monks to go learn how to read Latin and all sorts of stuff. So that way their monks and their priests could, go out and spread the word and actually know what the heck they're reading. But he became a monk, and he studied pea plants. And of course, the famous thing is he basically went out one day and he studied pea plants. He looked at wild peas and cultivated peas, and they made this funky hybrid in the corner of his pea garden, in the monastery's pea garden. And he was like, woo. And he was already really interested in genetics to begin with. Not that he knew it was genetics. They knew it had something to do with inheritance between father to mother. But they didn't really know what exactly was going on with that. Interestingly enough, he was alive around the same time as Darwin. These people never met, never talked. Actually, Mendel's work got lost for years until somebody found it again. And then we said, oh my God, he was talking about DNA, but he didn't know it. He called it factors. He called genes factors. He knew they were factors that was coming from parent plants into the offspring plants, and he came up with a whole bunch of math, and he worked with 33,000 pea plants in his lifetime to figure all this out, to do all this math. So that's why he's considered the father of modern genetics. However, recently, I just discovered this myself, and I've got to do more research to educate myself. Recently, a lot of his stuff about dominant and recessive and everything you got, beaten with in biology, in high school biology, if you took that. Yeah, guess what? It's all coming to head that none of that is actually that simple. But on the flip side, it gave us a launch off point to learn all this stuff. So I wouldn't poo-poo his work, because if it wasn't for his work, it wouldn't have given us an in onto how DNA possibly works and our modern understanding of how DNA and genetics works. And a lot of our understanding derived from him looking down and looking at this mathematically, which was really great. But now we're starting to overturn a lot of his what he's got, especially the dominant recessive thing, because it's actually a little bit more complicated than that. So we hats off to a man who basically just messed with pea plants and math and figured out things that people couldn't really grasp until we refound his research and said, oh my God. Crazy thing is, people used to, like I said, Darwin was his contemporary. Darwin used to bug everybody, or not Darwin, but people used to harass Darwin constantly about inheritance, and he didn't know anything about it. Just because he came up with natural selection does not necessarily mean, it's kind of like in movies. The scientist somehow knows every single field there is. We don't. We have specialized niches ourselves. So you walk up and talk to a general biologist like me, and even I have to go sit down and do some research on, people have thrown me with questions. I'm a generalist. I'm not exactly a specialist. I know certain things, especially about mammals, but that's, like if I have a question about botany, I'm going to go find a botanist and talk to them. And it's the same thing with Darwin and Mendel here. Darwin was into, how do species change over time, which does lend itself into inheritance big time, especially today. But unfortunately, people kept bugging him back then, and he was just like, I don't know. And unfortunately, he did get frustrated enough to say something that was completely and objectively wrong. And a lot of people clung to that for years. And that's where we get a lot of misconceptions out of natural selection, out of, stuff, until we found Mendel's research again. And it was funny. because Darwin actually had Mendel's research in his library, in his personal library. But back then they used to seal books, and you had to like cut the jacket open so you could open it up and read it. And his Mendel's work was still sealed, so Darwin never read it, which is hysterical to think because everybody was harassing him about it. And Darwin was smart enough to probably to open it up. And if he had opened it up and read it, he probably would have been like, dude. But I actually have some interesting journal entries by Darwin. He was depressed quite often. He was not a happy camper, especially after he wrote his book. He was just like, look, I'm just pointing things out. Stop harassing me and acting like I'm evil. And poor guy. He probably spins in his grave like a rotisserie mat to this day. Anyway, so Mendel had 4 hypotheses. For each inherited character, a trait, like, my brown eyes, we each have 2 copies of the gene, one from each parent. So these are known as alleles. So I got like a brown-eyed gene from my dad and possibly a blue-eyed, green-eyed gene from my mom, because my mom's got blue-green eyes. And so does my sibling. So, and of course I got the hazel eyes, which is actually kind of a mix. And he didn't know about the mixing, thank God. That probably would have thrown him. We do. And we'll talk about that in a minute. So anyway, so when 2 different alleles occur together, one of them may be completely expressed while the other may not have any observable effect on the organism's appearance. Hence, one trait is called dominant while the other is called recessive. Dominant traits are always expressed. Recessive traits do not. Now, like I said, this part's come under fire recently. We're honestly looking at it more and more, and the more we look, there may not be any true dominant traits, and there may not be any true recessive traits. So, like I said, this is what Mendel figured out from messing with nearly 40,000 Ps. So, and we kind of, changed it. That's a beautiful thing about science. Change is good, but also change that has been challenged and talked about and tested as much as humanly possible, the scientific method. La 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 la. What? All right, my snake is now getting stuck on no, Monty, why are you being like this? Why can't you just be happy for everybody?
I am going on an adventure.
Why are you stuck on there we go. Bad. Anyway, so when gametes are formed, alleles for each gene in an individual separate independently of one another. And we've already covered that. That's called meiosis. So the gametes only carry one allele for each inherited character. And when the gametes unite during fertilization, each gamete contributes one allele. So that brings us to Mendel's 2 laws. The law of segregation that states that 2 alleles per character separate when gametes are formed. We already went over this. This is meiosis. How basically eggs and sperm are created. And then the law of independent assortment states that alleles from different genes are separated independently of one another during gamete formation. So in other words, it's an absolute, Mother Nature does its best to completely scramble everything up to make it so we have as much genetic difference as possible, so that way we're a healthier species. However, keep in mind, between you and me, we only have 0.4 difference between each other. Yeah, that's just you and me, and we're not related in any way that I know of. You're not related to me, I'm not related to you, but if you're looking at genetically speaking, we pretty much are 99.6 identical to each other. Yeah, that's how close we are. Yeah, when you're watching Maury and stuff like that, or if you've ever watched Maury and they go, "You are not the father," or Jerry Springer, or wherever the freak that started. Yeah, that's actually really, really not that clear cut. The show makes it. Because certain populations are very, very much related to each other. Like if you come from a small town and, the small town's been only, just enough to keep it variable, but at the same time, how everybody knows everybody else. And I'm not saying Hendersonville. Hendersonville's got Asheville next to it. We got a lot of mixing going on around here. So anyway, I'm just saying, we're actually more related than most people think. So FYI, we are very related as a species. However, the genetic difference between us and Monty is pretty big. I mean, we've got some similarities, not physically, obviously, because we're not long tubes without legs and no ears, and have the brain of a pea, although sometimes I feel like I have the brain of a pea. Anyway, so stuff you need to know. Human body cells contain 46 chromosomes. We got 22 pair of autosomes, 1 pair of sex chromosomes, XX for us ladies, XY for you guys. Interestingly enough, just a random fun fact, birds are backwards. I don't know why. I don't think Monty's backwards, but birds are backwards. Males are XX and females are XY. I don't know why. Just random fact for you there. Body cells, or somatic cells, are called somatic cells. Sex cells are gametes. We talked about this earlier. Homologous chromosomes. So our 1 pair of chromosomes are similar in size, shape, and genetic content. And we map these suckers out, and I'll show you in a minute. It's called a karyotype when we map these out. And I've had to do it. And people still have to do it. We still do karyotypes to this day, even back when I was pregnant with my son. And then my second pregnancy that didn't go so well, they did genetically test. So, yeah, and you make a map of it all, and I'll show it to you in a minute. So anyway, diploid means you have the full number of chromosomes, and this is basically the number you should have in all your somatic cells, which is 46. You get one half from mom, 1 half from dad, 23 from mom, 23 from dad. So it's represented as 2N, or, yeah, uppercase or lowercase. It just depends on how your book wants to show it to you that day, or your professor. Haploid is half the full number of chromosomes in a gamete. Sex cell, egg, or sperm contains only 1/2 the chromosome in each homologous pair. It's usually represented just N, and it's 23 in us humans. You know, Monty, let me just see. I'm going to see how many chromosomes a ball python has. Let's look it up. How many chromosomes? Wow, this is weird. It says, how many chromosomes do humans have? Do dogs, do cats, do potatoes, and do chimps? It's funny what people look up. But humans is at top, so there you go. Does a ball python have? Okay, there we go. They have 36. Huh. Hey, Monty, you got 36. Anyway, not that he cares. So he's got 10 less than we do. Anyway, so in science, homo means same, whereas hetero means different. That's how I had to keep the high schoolers together on that one. It was just like, yeah, same thing, with it being June and everything. Yeah, definitely, same sex, different sex. However, you have to remember it. I don't mind. Just as long as you remember it. So homozygous means basically you get identical alleles. So say I got, brown-eyed allele from my dad and brown-eyed allele from my mom, which means I'm going to show up with brown eyes. Again, the dominant recessive thing is in question these days. But basically you get identical alleles on a trait from both parents. And anyway, eye color is not a good example because it actually merges. Like I said, I've got hazel because I know my dad's were kind of hazily and my mom's are green, and I've got that green flecked hazel. So there you go. It's more of a merger, really. Anyway, heterozygous, which I'll get into what that type is in a minute. So heterozygous means you get 2 different alleles for a singular trait. So, actually a good example of this is earwax. I know this is insane to talk about. One of the few things in the human body that's Mendelian genetics currently, until somebody corrects me, is earwax texture. I know. There's you either have wet earwax or you have dry earwax, and there's no real in between on that. So I've got really wet earwax. I'm constantly, if I don't clean it out, it drools out of my ear and drives me insane. So personal information. I'm filled with it. Anyway, so I don't know about you. No offense, I don't want to know. So basically, hetero means you've got an allele for dry earwax from dad and an allele for wet earwax from mom. It means you got 2 different. And genotype is the actual genetic makeup of an organism, is what your DNA you're carrying around says. Phenotype is what actually shows. Like, for instance, I've got wet earwax, and I forget which one's dominant, which one's recessive. I should look that up too. Because I know I've looked it up in the past and it just isn't occurring to my tiny mind right now. Sorry about that. I'll look it up later. Anyway, so, that was coded from the genotype. So the phenotype comes from the genotype. Now, the genotype is usually what you also, randomly pass on to your kids. 50% of which you have is passed on to your kids. The phenotype is just what shows up. Wild type is an allele. Now, this is weird, and this is some interesting naming, but wild type is an allele that's found in most common in a population. For instance, brown eyes. The majority of humans on this planet have some flavor of brown eyes. That's the wild type. I know it doesn't seem like you should name it that, because it's like, well, if it's the wild type, shouldn't it be wild? Shouldn't it be like weird and rare? But no, it's the actual opposite. It's what most people have. Weird, I know. I didn't name these. Once again, welcome to Miss Royal didn't name these. So, a mutant, this is an allele that's changed from a wild type or is uncommon in a population. For instance, people who have albinoism, or anything else that kind of is very much different from everybody else in a population. So, a mutant, and I don't mean like that in a bad way. I don't mean like cancer mutant, and I don't mean like, cool magical, well, not magical, cool powers mutant. Darn. Cool powers, but we don't get those, do we? No. So here comes the Mendelian genetics. Like I said, I'm just gonna touch on this now. You will be doing some monohybrid crosses. So I'll probably go over how to do those right now, because you will get a few, and I think I fixed them. The interactive worksheets you have, I swear to God, I think I fixed those, but that doesn't, because a lot of people were having problems with those last semester, and I thought I fixed them all. So if you're having problems with them, let me know, because there was one that kept showing up as wrong, but I thought I went in and edited it and fixed it. I might go back and check that before I pop it for you. But anyway, so say we have a trait with the letter C. Remember, we usually use the letter of the dominant trait, and I don't know why I used C's, because now they all look the same. I should have switched out and used like something that looks different. Like B's, you can definitely tell the difference between B's or D's. I should go back and fix this. Anyway, so homozygous dominant is capital. Heterozygous is always going to be big little. Always, 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 always. There's no other type. And then homozygous recessive always means lowercase. So a monohybrid cross is basically to use a grid. It was made by a guy named Punnett. Reginald C. Punnett. There you go. That was his name. And he was like huge into researching Gregor Mendel. And he came up with this fun thing to do that if you took biology, I assure you, you probably know, because either your science teacher beat you with it or not. I don't know if they'd beat you with it or not. I've been hearing now that you spend one day on it in class and move on, which is sad, because my genetics teacher just slapped me around with them. But I guess that was back in the day. Anyway. So test cross, by the way, is if you want to figure out, like, one of the parents is a recessive individual in a certain trait, and you want to figure out what the other parent is, and then you kind of look at the kid results. Now, keep in mind, when you're looking at these Punnett squares, the equals 100%. So each one of these squares equals 25%. And the whole thing together equals 100%. But this is probability. It doesn't mean for surezies. So for each one of these squares, you have about 25% of whatever is in there being the trait that shows up in your kit. That doesn't mean you're going to get one of each. It's kind of like one of my coworkers, former coworkers, great guy. Him and his wife had kids, and they had 3 daughters, and they tried one more time for a son, and he had another daughter. And it's a 50-50 shot, because if you go back with X's and Y's, and I can show you that. Let me go to brain. Let me go ahead and open up where is it, Monty? Where is it, Monty? Jamboard. There we go. So I can draw all over the no. So recent Jamboards. Open up. Boop. Open up a new one. Let's do it. So anyway, so for instance, I'm just yapping about, X, and X, Y. which is males and females, if you go ahead and make a horrible square. Sorry guys, doing this with my finger is not terribly good. I'm like, make sure I'm getting this. Okay, good. I just wanted to make sure I'm getting this. So again, if you put like you put one down the side, it's always the left, and then you put one across the top. Like we'll put the guy across the top here, and then you go ahead and you cross them. So you bring your X over and your X down. So you should have 2 letters per thing. X down, X over, X over, Y comes down. And as you can see, X, Y, there you go. That Y is terrible. Why is this terrible? It's so, no, that looks like an X. That was bad. There we go. A little better. All right, so forgive me drawing with my finger. It's not my strongest talent here. Anyway, but as you can see, no matter what you do, you got a 50-50 because like you said, did a Punnett square equals 100%. So you got 2 squares, 25. You can add it up to 50, add it up to 50. So you got 50-50 shot of having a girl or a boy. And interestingly enough, it's the guy that actually, and we'll get into this more in the next lecture, or last lecture. But you'll see what's going on is because the guy's got the Y and that causes you to be a guy, you've got a 50-50 chance. It's the guys that are actually in control of whether you have a boy or a girl. Not that the guys can control it in any way, form, or fashion. It isn't like you guys can sit there and go, wise, and that's what your sperm produces. It's just not how that works. You have no control over this. It's just 50-50, but it is your fault because you've got the Y's. We've only got X's, so we can only make more women. So it's kind of freaky. So anyway, so there you go. Craziness. So yeah, which brings me to a funny story about King Henry VIII a little later that I'll get into a little later. But that's basically how you do a Punnett square, and it shows you, you go 50-50 right there. So anyway, and so test cross is, again, going back to this. If you got a parent that's recessive in a certain trait, and you breed it with a parent that you don't know, depending on how the kids come up, you can usually you can kind of work backwards to figure out what the other parent is. That's called a test cross. It's useful in like plant breeding and whatnot. I don't see it too much when we're doing breeding with mammals and things, but keep in mind, we've got people that have been breeding mammals we've got dogs from wolves, for goodness sakes. We did a crazy job doing that. And cats, I swear to gosh, they domesticated themselves. If you look back at the history, they came on later down the road and just decided to waltz in one day and saying, "Hey, ancient Egyptians, I am fabulous. I am cute and adorable. So are my kittens. And we'll eat all the rats out of your granaries." And the ancient Egyptians went, "We love you." And then they got on ships and got all over the place. Interestingly enough, a cat on a ship is a sign of good luck for the ship. Isn't that weird? So cats on ships are actually good luck. If you're a ship person, you probably know that. I'm just one of those people that likes learning about shipwrecks all the time. But I'm not ever going to go to one because I'm afraid of the ocean. I swear that thing's going to get me one of these days. That's why I live in the mountains. Stay away from the ocean. It's scary. Anyway, so dihybrid cross is when you use a larger grid to figure out how 2 traits might come out between the offspring. Use FOIL. I am not going over this too much. I'm just showing it to you that, yeah, crosses can get bigger and crazier. But that's also the reason for computers. You can actually make a trihybrid cross too if you want to get really crazy with the math. I don't feel like that, so let's not. Anyway, and it's not something you definitely need in this class. Like, if you were taking a genetics class, yes. In this class, no. Anyway, so pedigrees. Pedigrees are still used. Basically, it's how we want to see a trait travels through a particular family, bloodline. Also, so it's a map of inherited traits. And squares are guys, circles are girls. Us girls do feel circular. Anyway. And if they're carrying a certain trait, and it doesn't have to be a good trait, it could be just looking at brown hair through a family, for goodness sakes. You can do it for almost anything. Or just blue eyes through a family, or any kind of thing like that mole on your left knee. So I see how that runs through the family. Like, for instance, I've got a notch in my ear right here that my son has the exact same notch on the exact same ear. Anyway, I don't know if, like, that's on my grandparents or something. I have to go stare at them. Anyway, so, and that could be something. But we still use pedigrees to this day. Like when I was mentioning in the chapter 23, when we were talking about the guys and the ones that would actually start off female and not turn into a male until they hit puberty when their testosterone really took off. We actually have gone in and figured out the genetics by first starting with sending scientists in there studying the genetics of this to set up a pedigree to see how it runs in families. So that was one of our first tools, setting down, trying to figure out that whole process and figuring out, how exactly this happens in a population and how much is expressed and how much it's showing up. So pedigrees is one of our first tools going in to try and figure out how things run through an area. So pedigrees is actually one of the few things that, if your science teacher taught it to you earlier, guess what? We still use that today. So that's not one of those things they've taught you and then you're like, oh, I didn't need that. Why didn't you teach me taxes? Which I have a whole rant about that if you want to hear it, but that's later. And not at all related to this class and will not be said in this class. But I have a whole rant about that being a former high school teacher. It's not us teachers, I assure you. We would love to teach you about taxes and other things you actually need to know. But that's another rant for another time. So anyway, karyotype. So again, as I was mentioning earlier, we take all the, basically what we do is we fluoresce them. We take a snapshot on it with one of the cells being in the middle of mitosis, and then we basically cut them out and glue them together. Or now we do it with a computer these days, so it's a little bit faster, but basically that's what we do, is we take, a blood sample, find somebody going through mitosis, fluoresce the DNA, and then everybody who's similarly shaped, we pair them together. And doing this usually lets us see if there's anomaly in any of the chromosomes, which like polyploidy, which we're going to talk about more in the next set of this last lecture. That shows us like the trisomies, especially trisomy 21, which is Down syndrome, trisomy 13, which unfortunately is like one of the major things they test for when you get pregnant, because if your baby has trisomy 13, there's just unfortunately it is not a viable pregnancy. It will not go to term. It will either die usually way beforehand, and it's not cool. It's not fun. Look it up sometime if you want to be depressed. But anyway, it's amazing how many babies are born every day in every way and happy and healthy, considering how much can go wrong and how much actually doesn't, is actually shocking. To me, anyway. It's just to me. So, and dominant traits are always expressed over recessive traits, unless we've got some different flavors going on. And even this is under fire right now, so we're just going to go with this for right now because that's what the book says, but I'm just saying current research says, and until they write that down in a book, I'm not going to beat you with it. I'm just saying, hey, look, science is changing on us right now. That's science for you. Because if science didn't change, we'd still think the moon was cheese. Actually, I don't think we ever thought the moon was cheese, but, just in case you were curious about that. So anyway, so when the traits blend together, we get incomplete dominance. And you can see this in snapdragon flowers. They love talking about snapdragon flowers. Basically, if you take a white snapdragon flower and a red snapdragon flower and cross-pollinate, you will get seeds that do nothing but make pink. This is where we get a lot of the incomplete colors. You see it in animals a lot, especially fur color. If you get a white cat and a black cat together, you'll get black and white cats and a whole bunch of gray cats. And so always spay and neuter your cats because they breed like crazy. I know. I had to rescue 3 of them and they live with me now. Isn't that right, Cassandra? Yeah, she's up there up on the tower right now staring out the window and not at me. The tower's next to me. Anyway, that's why they get occasional zoomies and run around on me. Anyway, so that's incomplete dominance, where basically the two traits mix, kind of like paint mixing. That's how I see it. Codominant, so remember when I talked about blood and you all got really kind of like, what? And I was like, yeah. And then you were like, how do you do a Punnett square again? And I was like, and I dropped the ball on that. Yeah, I should have done that better. Anyway, so codominance is basically when two dominant traits both express at the same time, they don't blend. So this is where we get the fun antigens and antibodies. So remember the phenotypes, and I don't understand why this book doesn't explain this then, but does explain it in this chapter, when the two chapters are like completely removed from one another. I need to fix that in my own notes. Anyway, so both A and B blood types are dominant over O, which is, interestingly enough, even though O is the recessive type, O is actually one of the more prolific of the DNA type, of the blood types. AB is actually kind of rare. But AB is a universal receiver. They can get everything. O is not. We're a universal giver, which is why the Red Cross calls me at least twice a week, if not 3 times a week, to drive me insane. Because they know I have, hold on. I have O type. Positive. O positive. That's what I was looking for. I was looking for positive. And my brain kept going, O plus. I'm like, that's not how you say it. You ever have your neurons just keep kicking out the wrong word and you're like, no, that's not the word, dude. Anyway. So anyway, you got AA and AO. That gives you basically A blood type. So you can either have A from mom, A from dad, or either or you can get A from one of your parents and O from one of your parents, and that's how O usually gets around. He's recessive, so you show up as as A blood type, even though you're carrying around a recessive gene for O. Then B, same thing, BB, you can either have B from mom, B from dad, again, B from one parent, O from another parent. And then AB can only be AB. That's it. If you get A from one of your parents and B from another one of your parents, it doesn't matter which, you've got AB blood. You've got AB blood. And then last but not least is O. And the only way you can get O is you get one O from mom and one O from dad. So me being O positive, I definitely got one from my father and one from my mother. So that's why I'm O positive. I don't even know what they are. I could probably text my mom. Anyway, my son is also O positive, which means the O, definitely one of them came from me, because that's the only thing I can give, because I'm both of my alleles for this are O. So I can only give O's to my kids, because it's a 50-50 shot, but it's a 50-50 shot of the same thing. So O-type people can only give off O's to their kids. So mine's pretty easy where one of his O's came from, and obviously the other O had to come from my husband. So he's O something, and he's too much of a big fat chicken when it comes to blood. No offense. I love my husband dearly, but he does not like the sight of blood. He doesn't faint at it, but he gets lightheaded and woozy, so he will never be in the medical field. Oh, well, that's okay. I love him for his nerdiness, his nerdy mind. That, and he will sit there and complain about nerdy things with me, like video games and other things that amount to nothing, but we like complaining about them. Anyway, I hope you too find a significant other that also enjoys complaining about the same things. Anyway. So, like I said, yeah, my son's O, I'm O. I want to say my mom was A, but I'd have to text her. I'll text her right now. What blood type are you for educational purposes? No. There we go. Boop. All right, there we go. She'll get back to me. Anyway, So, single expression of single genes. So, this is pertinence and expressivity. So, a genotype is completely pertinent if not all the individuals inheriting express the phenotype. So, in other words, if not everybody's got it, is showing it, then it's probably incompletely pertinent. A genotype is variable expressive as if expressed to different degrees in different individuals. So this is like skin color and stuff. So, skin color is definitely not incomplete, and it's definitely not it's something else, and we go over it a little bit later, but yeah, it's everybody, trust me, gets a different, we're also seeing the variable expressive, especially with the spectrum. No matter what spectrum it is, there's a Down syndrome spectrum. We've been learning more and more, there's more spectrums than we actually originally thought. We either thought you have it or you don't. And then we were looking at Down syndrome and we're like, well, there's a whole range there, because there's a lot of people that have Down syndrome that, they're somewhere in between actually fully functional and can live a pretty much normal life. And then there's people that need to be, in a facility that takes care of them for the rest of their life because they cannot take care of themselves. And there's a ton of people in between. Okay, my mother is A negative blood. That's interesting. So I knew there was an A floating around somewhere. So, my mom was A negative, so she definitely had to have an O. So she had a recessive O gene, and that came to me. Now I wonder what my bio dad is, but I don't talk to him, so I don't know. Anyway, pleiotropy. So this is a disorder that has like several symptoms and several substeps which may affect individuals, like Marfan syndrome. That's a whole spectrum too. This is a result of gene production that is part of more than one reaction organ structure. So we've been finding this out more and more and more as well, that it usually takes more than one gene being turned on and off in certain ways to get some of these results, whether they be a disorder or just regular phenotypical things that show up in the population. And it used to be, when we did the Human Genome Project back in the 90s, and that was a huge thing when I was a kid, was, we're doing the Genome Project, we're going to map every gene and everything on the human genome. It actually, they did it faster than they thought. They thought it would take 10 years. They did it in 7, and we're shocked because we didn't have that much. And we were like, because we used to think one gene, one thing. And we used to think we were like, not spark plugs. Yeah, I'm trying to equate it to cars. You go in, you pull out the plug. My brain once again, my brain's kicking out spark plugs, and I'm like, that's not spark plugs, dude. It's like it's like on your house too. You blow fuses. Thank you. Sorry. Fuses. So yeah, if you can pull a fuse out and put in a brand new fuse, that's what we used to think. We used to think gene therapy was like, oh no, you've got, something. Well, or like, here, you've got diabetes. Well, that means you've got a bad gene. Let us go in and pull out the bad gene and put in the good gene and no more diabetes. But unfortunately, that's not how that works. It was actually way more, then we found out, because we don't have as much as we thought, it's because several genes turn on and off in certain combinations to get what we've got. And even then, they're turned on and off not because of just how we're born, but because of our environment now, too. And that's where I was talking about that other thing with going up to space, and things get turned on and off on a daily basis, depending on what we eat, depending on if we exercise or not, depending on what we do. So it's a lot more varied than that. But now we're kind of making the full circle and going back to, well, we can pull out certain things and put in new things, and that should help again. So gene therapy was dropped for quite a while because we used to think it was plug and play. It's not plug and play, but now we're coming back around to the thought of gene therapy again, especially with cancer patients, because now we're learning that even cancer is kind of specialized to what's going on with your DNA alone. Even though 2 patients may have lung cancer. Let's take lung cancer, unfortunately, claimed to my dad, who I consider my real dad. My bio dad is gosh knows where, but my real dad, who I consider my real dad, he passed away of lung cancer. And, his lung cancer was, even though it's classified the same as somebody else with lung cancer, it was different because cancer is exclusive to that person because of our genetics. And that's the problem why we can't have a blanket medication for every type of form of cancer. And that's why I get mad when I get those conspiracy theory people. And like I said, I'm not knocking them because I've got my own weird conspiracy sometimes too, put the tinfoil hat on with my own silliness, but I keep that to myself. But it's when somebody's like, oh, the government is withholding the cure for cancer. And it's like, yeah, but the problem is cancer is complicated. And just because two people have lung cancer doesn't mean it's the same, because it's different between those two people because of their genetics and how they got it and all this other stuff. So it isn't exactly we can't really find a bullet to fix everything. We have to figure out, a lot of different things. And that's actually why we're now looking at gene therapy again, because now we're tailoring medication to that person's genetics and genetic profile, what will work best with their genes to defeat their cancer. So that's, again, some of the interesting things that are coming about this. Sorry, I didn't mean to go on that kind of a rant, but I just think it's really fascinating how we're kind of going full circle on what we originally thought, and then we're coming back around again. Looking at it a second time, even though we're not plug and play, we're getting better. So, and then her genetic heterogeneity, which basically refers to a phenotype resulting from a mutation in more than one gene, like clotting disorders. There's actually a lot of different types of clotting disorders. Hemophilia is not exactly one of them. I mean, no, it is one of them. I'm just saying there's more than just hemophilia, which is, famous because it goes down through the English royal family, because, back then they thought, if cousins marry cousins, that will just make you more royally, except it made you more genetically awkward. So, and if you think the royal families of Europe were bad about that, look it up sometime about how many genetic conditions Tutankhamun had, because his sister with his mother was his father was his uncle. Trust me, the ancient Egyptians had sisters and brothers marrying each other and having kids constantly, because unfortunately their own tales said, yeah, that was okay to do, and it's not. We know that now, but back then nobody knew that in the days of ancient Egypt. So yeah, crazy. So yeah, Tutankhamun, we've done some of his DNA, what we could find, what was left, to try and figure out who his parents were and everything. We actually were testing a lot of the mummies that we've got of what we think is who we think we have, because a lot of them got jumbled up for many, many reasons over the many, 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 many moons. So we were doing genetic testing on all the famous mummies in the Cairo Museum, and a lot of that stuff was coming out. It was really fascinating, because we were trying to figure out who's your uncle, except mummy edition, except we found out a lot of them are their own uncles. The song where I'm my own grandpa, it's like that, except real. And that's why Tutankhamun had so many genetic problems. Oh God, he had so many. Look it up sometime. Oh. So anyway, so again, we've got multifactorial traits. This is basically what eye color is right here. It's molded by more genes plus their environment. They're called multifactorial or complex traits. So as I said, your environment does actually turn your DNA on and off. And even when you're pregnant with a kid, or, somebody pregnant with a kid, the kid developing inside of you is also developing their own gametes at that time. So anything you do may have affected how your grandkids are coming out, if you have grandkids. Like, for instance, when I was pregnant with Wesley, things I may or may not have done could have turned on or off DNA I'm not aware of, and that could be reasons why in the future, my grandkids come out the way they do. So yeah, you actually have your grandparents actually had, or especially your grandmother, had a lot of influence on how you were made, because your parent was, in their womb when they made their gametes. Isn't that insane to think about? So yeah, instead of blaming your parents, go blame your grandma. No, I don't mean that. No. I don't want to get hit by a shoe. Anyway, moving on. A trait that is determined by more than one gene is called polygenetic. So examples of this: eye color, skin, height. Interestingly enough, the human species has been getting taller over time due to nutrition and just height reasons. So if you ever go to like older houses, especially up in these thar hills around here, you'll notice in older houses, like colonial houses, some of you have to stoop to get in. Yeah, that's because it's usually, the frame was usually built, not standard back then. It was kind of like built to the tallest person in the family. So yeah. Interestingly enough, get this, everybody's got it wrong when it comes to Napoleon. Apparently he was not short. He was completely average for his height. He was not short. So he was like 5'6". But he wasn't short. Everybody thinks Napoleon was short. He wasn't, actually. For his day and age, he was actually kind of tall. So, FYI, Napoleon wasn't short. I mean, compared to a basketball player these days, yeah, he was short. Or Andre the Giant, or The Big Show. Sorry, did I just show that I used to watch WWE? Probably. Is The Big Show even wrestling anymore? Anyway, moving on. I don't know. I haven't been paying attention. So anyway, here's a really good one on heredity. So it goes through earwax just like I did. Good stuff right there. There is another one right here. Highly recommend. I think I threw at several people when they messaged me about not understanding Punnett squares. It's right here. It's a beginner's guide to Punnett squares. He does an amazing job. He also goes into how to do a dihybrid cross. You don't need to know that pain unless you want to torture yourself. I'm not even going to touch that with a 5-foot pole. You will be having to do some Punnett squares though. So please, please, please review how to do a Punnett square with this video, or harass me and I'll make a video. Trying to think. Yeah. So highly recommend, because you will be doing some Punnett squares with the work. And like I said, I need to go check that. All right. With that said, I'll leave you for now, and I'll see you in our next and last lecture for this semester. See you. Bye.